In this video, we will discuss about a strategy that can help in spatial detection of GlycoRNA. So these GlycoRNAs are the one are those RNAs that contains the glycan structures that is shown here. That these are the RNAs and it contains the glycan structures. And I have already made a video about the GlycoRNAs and how they are being detected for the first time. I have discussed that paper in details. You can go through the reference or you can check the I button. From there you will be able to know how these glycoRNAs are being detected using mass spectrometry. In this uh, video we will discuss a different strategy to detect glycoRNAs, that is using fluorescence. So this is the overview of that strategy, in which they have this aptamer. Aptamer can be specific to the glycan structures, that is it can be specific either to the sialic acid or any other monosaccharides. And apart from this there are two different probes. First one is the glycan probe and next one is this RNA binding probe, and both of them are connected using this connector. So combining all this it will form a uh, uh, combining all this it will form a it will form a it will form a fluorescence indicators, that whenever that is whenever this sensor detects any glycoRNA specific to this aptamer, it will give a fluorescence indicators. And in order to prevent the false negative, how it will work is that it has two different uh, probes, that is glycan probes and RNA binding probes, and along with this there is the aptamer. So this aptamer so whenever all these three conditions are satisfied, then it will give a signal, and it is shown here. That is when the aptamer specific glycan structures are present on the RNA, it will attach to the glycan probes, and along with that, if these glycan structures are present on the RNA, this RNA binding probe is also been attached, and because of this connector, then it will start polymerizing using this PHI DNA polymerase. And this along only this polymerization will not give a fluorescence indicator. In order to get a fluorescence indicator, what it will does is this aptamers needs to be in close proximity to this newly translated newly translated uh, hybrid sequence, newly translated sequence, or newly translated this DNA structures, this DNA sequence. And when these aptamers and these newly translated uh, DNA structures are in close proximity, then it will give a fluorescence reporter, and that will provide us the signal that this kind of RNA that contains aptamer specific glycan structures are present. So this close proximity will help us avoiding any false positive signals. And this is the figure in which they have tested this their new reporter, which they called as ARPLA, and using different controls. So first they have used their wild type ARPRA that will gives this Alexa fluoro 647 signals. In this case, they have used the for this studies in this figure for this figure they have used the aptamer that is specific to the sialic acids. And when they don't use this aptamer, since the aptamer is not present, only only the RNA binding region will be translated, and that will not give any signals that can be seen here. And after this, there is without rush, or with in this case without DNA binding probe, the this region has all of half of the regions will not get translated, or there is no region will get translated. So because of this, it will not give any signals. And without the connector also, it will not get translated and it will not give any signals. Next thing, using the glycan probe with different DNA sequence, or we can say scrambled sequence, then also it will not give any signals, because this DNA sequence will not match with the RNAs, or will not match with the RNAs that are interested for which this ARPLA is designed. So this is the quantitative analysis of this figure is shown in this panel B. We can see that the signals from ARPLA is significantly higher than the other control condition. And next studies that they did is by inhibiting the glycosylation using different inhibitors. First they did not only with the glycosylation but also with the RNA. So first they did not inhibit without any cells, without any treatment gives the normal signals, and next in this case they used their actual ARPLA reporter. And when they treat the cells with RNA-Z, that leads to the degradation of RNA. They see the very minimum signal, and similarly when they use the RNase T1, they see very minimum or negligible signals. And quantitative analysis shows that without treatment has the significantly higher signals compared to the with treatment. Next, they treat the cells with different glycosylation inhibitors, such as this K5-nuclease. It leads to the N-swinizonine. Both of them leads to the inhibition of N-glycosylation. So they will not see any signals because sialic acids are mainly produced by the N-glycosylation, so they will not see any signals. And next they use this neuramidase or pen-neuramidase to treat the cells with to cleave the sialic acids from the glycan structures. In this case also they do not see any signals, and with PNGSF also they do not see any signals. However, when they treat the cells with O-glycosidase, they see many signals because N-glycosylation still have the sialic acids present, so it will give the signals. So this studies or this experiment validate their spatial detection technique of ARPLA that and that their ARPLA will work only when the aptamer specific glycan structures are present, and also these actual DNA structures, actual DNA sequence is present. So and it will not give the chances of false negative signals are very low because they do not see any signals in the case where they are not expecting any signals. So, and the last one is this. I forgot to discuss about this panel. It is the quantitative analysis of this fluorescence images. In this case, we can see that without treatments are similar to the O-glycosidase because O-glycosidase has minimum sialic acids in the normal healthy cells. So I hope you got an idea how this GlycoRNA can be detected spatially using this ARPLA detectors, fluorescence detector. And if you need to understand more about this spatial detection of GlycoRNA, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.